Hi, welcome to Rocky Biochem series. In today's video, we will be discussing glycolysis. Please, if this is your first time of watching our video, kindly subscribe for more important videos of some key topics in biochemistry and molecular biology. Glycolysis is a combination of two words, glyco and lysis. Glyco means glucose and then lysis means breakdown. So literally, glycolysis means glucose breakdown to produce pyruvate. Now let's look at some key facts about the glycolytic pathway. So the pathway is a catabolic process, meaning it involves the breakdown of a complex metabolite to produce a smaller one. So glucose is our complex metabolite here, and then the pyruvate is a simpler one. And it's also an anaerobic process, that is, oxygen is not a requirement for the process to take place. And then the cellular compartment in which it occurs is the cytosol. As I said earlier, the starting material is glucose, and then the product is pyruvate. So the glycolytic pathway is made up of 10 enzyme catalyzed reactions. The first 5 reactions are called the preparatory phase, and then the remaining 5 are called the payoff phase. So the first reaction here involves the phosphorylation of glucose to produce glucose 6-phosphate, and the enzyme involved is called hexokinase. The enzyme uses ATP as a source of phosphate group donor. The enzyme's name can be derived from the reaction ongoing and then the substrate. So glucose is a hexose sugar, so that's why the enzyme has a name hexo. And then any enzyme that catalyzes phosphorylation of intermediates is called a kinase. So since glucose is a hexose, then the enzyme is called hexokinase. So after getting the glucose 6-phosphate undergoes isomerization to produce fructose 6-phosphate, and the enzyme involved is called glucose 6-phosphate isomerase. So as I said earlier on, we derive names of enzymes based on the reaction ongoing and the name of the substrate, or let's say the reactant for that particular reaction. So the glucose 6-phosphate becomes our reactant, and then the fructose 6-phosphate becomes our product. So the glucose 6-phosphate here should be part of the name of the enzyme. So since the kind of reaction ongoing is isomerization reaction, then we can call the enzyme glucose 6-phosphate isomerase. Now after getting your fructose 6-phosphate is also going to undergo another phosphorylation at carbon number 1 to produce fructose 1,6-base phosphate. Fructose 1,6-base phosphate, and the enzyme involved is called phosphofructokinase. Phosphofructo from fructose 6-phosphate, and as I said earlier on, any enzyme that catalyzes phosphorylation reaction is called a kinase. So phosphofructo then kinase. So after getting your fructose 1,6-base phosphate, that particular reaction too involves ATP as a source of phosphate group donor. ATP is a source of phosphate group donor. So after getting the fructose 1,6-base phosphate, it undergoes aldolytic cleavage by an enzyme called aldolase to produce dihydroxyacetone phosphate and glycerol phosphate. So the dihydroxyacetone phosphate is a ketone, is a keto sugar, whereas the glycerol phosphate is an aldose sugar. The next step of the reaction involves the glycerol phosphate intermediates as the substrate. So therefore, dihydroxyacetone phosphate cannot be used directly as a substrate. Therefore, it has to be isomerized to form another molecule of glycerol phosphate. So the enzyme involved in this process is called the triose phosphate isomerase. Triose phosphate isomerase. So dihydroxyacetone phosphate is a triose, and then glycerol phosphate is also a triose. So we are just isomerizing this particular dihydroxyacetone phosphate to glycerol phosphate. So since the name of the reaction here is isomerization reaction, and then the two of them are both triose bearing phosphate group, then the enzyme is called triose phosphate isomerase. So now, after getting your glycerol phosphate, then it will start the next phase of the reaction, that is the payoff phase. So the first reaction of the payoff phase involves the conversion of glycerol 3-phosphate to produce 1,3-base phosphoglycerate. 1,3-base phosphoglycerate. So this first reaction of the payoff phase is just an oxidation reaction. So the glycerol 3-phosphate here is going to be oxidized by using the NAD plus. So the NAD plus will take off hydrogen from the glycerol 3-phosphate to form the 1,3-base phosphoglycerate. One thing that we should take note here is that as the oxidation is ongoing, phosphorylation is also going on at the same time. So this time around, the source of phosphate donor is not ATP, but rather an inorganic phosphate in the cell. The 1,3-base phosphoglycerate is going to undergo dephosphorylation to produce 3-phosphoglycerate. We are going to remove the phosphate group at position number 1 to produce 3-phosphoglycerate. And the enzyme involved is called phosphoglycerate kinase. So anytime you hear kinase, I told you something that when the reaction involves removal or addition of phosphate group, the kinase enzymes are those involved. So after getting a 3-phosphoglycerate, here too, we used ADP as the source of the phosphate group acceptor to form ATP. So the first reaction that leads to the formation of ATP is the conversion of 1,3-base phosphoglycerate to form 3-phosphoglycerate. So after getting the 3-phosphoglycerate, the 3-phosphoglycerate undergoes isomerization to produce 2-phosphoglycerate, and then the enzyme involved is called phosphoglyceromutase. Phosphoglyceromutase isomerizes 3-phosphoglycerates to produce 2-phosphoglycerates. Now, the 2-phosphoglycerate is also going to be converted. It involves the removal of water from 2-phosphoglycerate to form phosphoenol pyruvate, that is PEP for short. Water is removed from 2-phosphoglycerate to form phosphoenol pyruvate, and the enzyme involved here is called the enolase enzyme. So the phosphoenol pyruvate formed then undergoes another round of dephosphorylation to produce pyruvate, and then the source of the phosphate group acceptor is ADP to form ATP, and the enzyme involved is the pyruvate kinase. So the whole of this glycolytic pathway, we have a very important reaction. That very important reaction is what we call the committed step. So the first committed step of glycolytic pathway involves the phosphorylation of fructose 6-phosphate to produce fructose 1,6-base phosphate. That is, the phosphofructokinase catalyzed reaction is the first committed step in glycolysis. When we say a first committed step, it is just a reaction which, when it is bypassed, the rest of the reactions proceed into equilibrium. So that is, if we can't bypass that particular reaction, then remaining of the reactions can't proceed. Now let's look at regulation of the glycolytic pathway. How the glycolytic pathway is regulated. So we have allosteric regulation. Allosteric regulation has to do with some enzymes having a site called allosteric site. So when some activators or inhibitors binds to these allosteric sites, they bring about regulation of the activities of the enzyme. So we have allosteric activators. The allosteric activators here are fructose 2,6-base phosphate and then AMP. So when the levels of fructose 2,6-base phosphate goes up, it brings about activation of phosphofructokinase enzyme, and then this brings about activation of the glycolytic pathway. And also, when the levels of AMP increases in the cell, it brings about activation of the phosphofructokinase enzyme, making the glycolytic pathway being favored. So AMP levels goes up when the energy of the cell goes down. That is when there is low ATP level. We also have allosteric inhibitors. They are ATP and then citrate from the TCA cycle. When the levels of ATP goes up, it means the energy levels of the cell is already high. So there is no need for breakdown of glucose to produce energy. So this brings about inhibition of the phosphofructokinase enzyme, bringing about inhibition of the glycolytic pathway. And the citrates from TCA cycle also serve as an allosteric inhibitor. When there is high levels of citrate, it means there is high levels of energy in the cell. So there is no need for additional glucose to be broken down for energy to be extracted from it. Then we have another way of allosteric regulation. This involves the pyruvate kinase enzyme. That is the enzyme that catalyzes the conversion of phosphoenol pyruvate to produce the pyruvate. So this particular enzyme here too is activated by fructose 1,6 bisphosphate, and it is inhibited by ATP and alanine. We can also talk about hormonal regulation. Hormonal regulation has to do with how some hormones bring about regulation of the glycolytic pathway. So, in the case of hormonal regulation, the hormone called insulin serves as an allosteric activator for the glycolytic pathway, as glucagon also inhibits the pathway. And then we have substrate level control. That is how some substrates also foster or inhibit the glycolytic pathway. So anytime there is high level of glucose in the cell, it activates glycolysis. That is why as any human being you eat, the first 40 minutes that glucose is abundant in the blood. That is when glycolysis is at its peak. And when there is low level of glucose in the cell, it inhibits glycolysis. Thank you very much for watching our video. Please don't forget to subscribe and share the link for others to also enjoy. Thank you very much.